Here is the last part of our prokaryotes lecture. And this is a pretty short part. Really all we're going to worry about here is basically just some naming things. This is on phylogenetics. And phylogenetics just simply means relating to relationships between organisms. How do different prokaryotes relate to one another? So we can divide out bacteria into several taxonomic groups. Taxonomic groups just mean naming groups or way we classify the bacteria. Those bacteria that are gram-positive, we call them the firmicutes. Those that are gram-negative, we call the gracilicutes. Some of the bacteria that we talked about earlier, like the mycoplasmas, remember, don't have a cell wall. We put those into the category tenricutes. And then those bacteria that are the archaea bacteria, they are kind of a group unto themselves, and they are classified as the mendosicutes. So those are our four major taxonomic groups that you need to know. Now, when we look at these different taxonomic groups, obviously there are different species within each of these groups. And let's make sure we're clear on the definition of species. A species means a collection of bacteria that have a similar pattern of traits. A species is a population or group of bacteria that share similar patterns of traits. You can also have subspecies, or what are sometimes called strains, or sometimes called types. And these are groups of bacteria within the same species, but that have slightly different characteristics. So I might, for example, have a bunch of a big population of bacteria that all are the same species, but within that species there may be a group over here that differs slightly from this group over here, and that one differs slightly from this group over here. Those are each different strains, or you could call them different types, or you could call them different subspecies. Later on in the semester, when we start learning about your immune system, we'll also learn more about something called serotypes. And these are, this is a way that we can distinguish different bacteria within a species based on how different antibodies respond to them. And a good example of this is there are, oh shoot, now I just blanked out on it. There are several, about 2,400 different types of salmonella bacteria. They're all within the same one species of bacteria, but there are all these different serotypes, 2,400 different serotypes of salmonella. Okay, now there are always going to be a few bacteria that don't really fit into any one category very clearly. So I'm going to talk about just a couple of them, and I'm only going to focus on a few that are pathogenic, meaning that they cause disease in humans. And the first one I'm going to talk about are rickettsias. Rickettsias are very, very, very tiny bacteria, and they are parasitic. In fact, they do not have the ability to survive outside of a host cell. They tend to be gram-negative, and they are spread by an arthropod vector, which is a fancy way of saying that an insect, in this case ticks, like wood ticks, spread rickettsia when they bite an organism. So here's a picture of a host cell, and all these dots inside of this host cell, this is the outside of the host cell, but all the dots inside, those are all rickettsias cells. So you get an idea of just how tiny these are. And there's one type of rickettsia that's responsible for causing a disease called Q fever. There's another version of rickettsia called rickettsia rickettsiae, and it's responsible for Rocky Mountain spotted fever, which is a disease that does affect humans. Another class of unusual bacteria are the chlamydias. Chlamydias are also parasitic. They are also small. This is a human cell here, and it has chlamydia inside of it, reproducing, such as you see these two kind of bubble-like looking things here. Those are two chlamydia bacteria. These guys do not get spread by a vector. In other words, they don't get spread by an insect, but they do get spread by sexual contact. So this is an example of a sexually transmitted disease. And last, very briefly, let me just mention again the Mendocicutes. Mendocic, remember, is another name for the archaea bacteria. These guys, recall again, do not have a peptidoglycan cell wall. They are not pathogenic, meaning they do not cause disease in humans. They still have 70S ribosomes like other prokaryotes do, but unlike bacteria, the other group of prokaryotes, Mendocicutes, or the archaea bacteria, are unusual in that they tend to live only in very extreme environments. They are what we call extremophiles. Phile, again, means loving, so they love extremes. Some of them are halophiles. Halophiles love salty environments. So, for example, if we look at the Great Salt Lake, like you see here, the water there is can have a pinkish hue to it. That pinkish coloration is due to the bacteria that are growing in the extremely salty environment. Thermophiles are bacteria that grow very well in high heat conditions, like in the thermal pools in Yellowstone National Park. And then methanophiles are organisms that grow on or near methane gas, like, for example, what you would see next to volcanic vents where volcanic gases are being released. And that's the end of our lecture on prokaryotes. Next up, we will be talking about eukaryotes.